Today we're going to talk about the blood vessels, and so we're going to break this lecture into two parts as well, since it is fairly long and it can be a fairly challenging topic. So our blood vessels are going to be part of your circulatory system. They are a closed system, so it's a system of tubes that are going to carry bloods to and away from the location, and it stays within those tubes the whole time. There's 5 main types of vessels that we have. The arteries are the vessels that carry blood away from the heart. The arterioles are going to be the smaller branches of the arteries. They're more muscular. And then we have the capillaries. These are going to be the smallest of them. They're going to be the site of where you have the exchange of gas and nutrients. And generally, they're going to connect the arteries to the veins. So after blood passes through the capillaries, it will move into the venules. They are less muscular than the arterioles. And then move into the veins, and it's going to carry blood back to the heart. So there are 3 layers to the vessels. You've got the tunica interna or tunica intima. That's the innermost layer. The tunica media is going to be the middle layer. And then the tunica externa is the outermost layer. So the main differences are in those layers, how we modify them in these different types of vessels, and they're just modified to be able to account for the differences in what they do. This is a nice picture here that shows the differences between them. So you can see here with a large vein, an elastic artery here, the difference in the size. And then you can see for the muscular arteries or the medium-sized veins, the differences. And then down here at the bottom with the capillaries. So your key differences in here are the size, and you can look and see the muscular layer is what's going to be the key difference for all of them. So your tunica interna or tunica intima is going to be the part that lines the blood vessels. So this part is actually exposed to the blood. Endothelium is going to be the simple squamous lining over the basement membrane. So that's going to be what's innermost layer here that you can see in these pictures. So it's going to act as a selectively permeable barrier, so some things will be able to pass through this. It will secrete chemicals that are going to stimulate dilation or constriction of the vessel. This is done to help control its size. It normally repels the blood cells and platelets that may adhere to it. That's an important thing. It's kind of almost like having a Teflon layer in there that can be turned on and off. But you want to have them not sticking in there and only allow it to happen when you want to form a clot. So that when the tissue around the vessel gets inflamed, the endothelial cells will produce a cell adhesion molecule that will cause the leukocytes to start to stick to the surface. So at that point, it's like it shuts the Teflon layer off and things can start to stick. This will allow them to congregate in the tissues where they are needed for their protection. So your tunica media is your middle layer. It's going to be smooth muscle, collagen, and elastic tissue. This is going to give the vessel strength and allow them to resist the pressure that's pushing against them. Vasomotion is changes in the diameter of the blood vessels. So this is something we want to have happen. This is brought about by changes in the smooth muscle. Again, it's a way of being able to control blood flow in an area. Then the tunica externa or tunica adventitia is the outermost layer. It's going to have loose connective tissue that will help to merge with the tissues of the neighboring blood vessels and organs. It helps to hold them in place and then provides a way for passage of the small nerves and lymphatic vessels. So the vasa vasorum are the small vessels that supply blood to the outer half of the blood vessels. So even though they're filled with blood, the blood vessels, tissues themselves need to be fed, and that's what the vasa vasorum is for. The blood in the lumen is thought to nourish the inner half of the vessel, but the outer half would need to have a little bit more. So this is a picture here again showing the different layers going from the lumen on the inside to the outside, so you can see the layers are just much thicker on an artery. And then upon dissection, you can actually feel a significant difference between the thickness of an artery and vein. You can feel the muscularity in an artery's walls. So here we have some varicose veins. So where you commonly see them on people is in the legs. They do occur other places, but you're more likely to walk around with your legs exposed than some of these other areas where you can have varicosities. So what happens with a normal vein is you've got the valves here that will close this off. It prevents the blood from having backflow in there. So this is blood that's returning back to the heart. You don't want it going back to the tissue, and that's what these flaps here, the valves and the veins, are doing. With the varicose veins, you've had them get stretched out, and now these valves are no longer effective at closing things off, and you can have blood pooling back down, which then just stretches them out even more. So they can occur in blood vessels other than the legs. So hemorrhoids are actually varicosities in the rectum. And no, you don't see people walking around with those because we just don't expose it, but they have them. So looking at the arteries in a little more detail, there's 3 layers to the typical blood vessel. So you've got the thick muscular to elastic tunica media. One of the properties of it is it has high compliance. The walls are able to stretch and expand in response to the pressure without tearing. So this is important because our pressure in our vessels is going to increase when we are more active. It needs to increase to supply the tissues. So functional properties of arteries are elasticity and contractility. Elasticity is because of that elastic tissue in there. So it will allow it to stretch when you're in a lot of pressure from contraction of the ventricles. So you're going to see more elasticity in the larger vessels. Contractility would be from the smooth muscles. This will allow these vessels to increase or decrease the size of the lumen. So they can decrease the size of the lumen for vasoconstriction, and that's used to be able to shift blood flow away from that particular area and send it somewhere else. So this is showing the different layers of the tissue under an actual microscope from a transverse section of an artery. So vasoconstriction decreases the lumen size, vasodilation increases the lumen size. So again, more comparisons between arteries and veins here. You can see size of the lumen being quite different here. So the arteries are the ones that are going to undergo the changes in their diameter. So elastic arteries are going to be your largest ones. So the tunica media has a lot of elastic fibers. So this would be something like the aorta that's going to help get blood away from the heart, the pulmonary and carotid arteries. So they'll expand during systole, and then they'll automatically recoil during diastole. This actually helps to lessen fluctuations in the blood pressure. So they can act as a pressure reservoir. Your muscular arteries are more about distribution. They're medium and smaller size. They can vasoconstrict and vasodilate. An example would be the radial artery. And then resistance arteries are the small arteries. This would also include your arterioles. They're going to control the amount of blood to various organs. So for example, if you are running, you're going to shift some of the blood away from your digestive system because digestion is not your focus at that point, and shift it to skeletal muscle. Meta-arterioles are short vessels that will connect the arterioles to the capillaries. So they have precapillary sphincters in there that are going to help to monitor capillary blood flow. So if you constrict the sphincter, it's going to reduce blood flow through those capillaries, helping to redirect the blood flow to other places in the body where it's needed. Anastomoses are where you have branches of 2 or more arteries supplying the same tissue connect. It provides an alternate route or a collateral circulation. So this here is showing the arteries and them being a pressure reservoir in here. So blood coming out of the ventricle, pushing out against the aorta and arteries to stretch them, and then them coming back to size. And then this is showing a capillary bed here. So you've got a meta-arterial precapillary sphincters in here. And then you've got your true capillaries. And then it will head over to venules on the other side. So with capillaries, you've got an endothelial tube inside of a basal lamina. They're the smallest vessels that are going to connect the arterial and venous blood. So with microcirculation, again, you're going to have flow from the meta-arterial through the capillaries into the postcapillary venule. The job of this vessel is exchange. So you can exchange gases and nutrients between the bloodstream and the interstitial fluid. The tunica media and tunica externa are non-existent in capillaries. So the substances only have to pass through the layer of endothelial cells in the basement membrane. That's it. So capillary beds will arise from just a single meta-arterial. So here you can see a capillary bed and it being just big enough for those single row of cells to fit through. So vasomotion is the intermittent contraction and relaxation. This helps keep fluid moving through them. And then you can have thoroughfare channels that will bypass the capillary beds. So these are your capillary beds here. Here is one of the meta-arterials. And then this is a thoroughfare channel. So we have three types of capillaries. So the continuous ones, the endothelial cell membranes form a continuous tube. So that's what we can see here. The fenestrated ones have fenestrations or pores. That's what a fenestration is. So you can see the little pores in the walls here. And then the sinusoids are discontinuous. They're wider and more winding. They usually have large fenestrations in there that allow proteins and clotting factors to get through. The blood cells can enter into the circulation here. They've got low pressure vascular channels. So you would see this in places like the liver, the spleen, the marrow, the lymph nodes. And here you have a lot of filtration to remove microbes, antigens, and old red blood cells. So here you can see the spaces are much bigger in these. So the capillary beds are organized into networks that are usually supplied by just that single meta-arterial. You've got that thoroughfare channel that will go through to the other side. What goes in and out is controlled by those precapillary sphincters. So they are well perfused. So when the sphincters are open, the capillaries get a lot of blood flow in there. And the blood can engage in exchanges with the tissue fluid. So exchanging nutrients, oxygen, carbon dioxide. When those sphincters close up, the blood is going to bypass the capillaries. So it can go through the thoroughfare channel to the venule instead. So these are all over the body, and perfusion is an intentional process, so it's not just automatic. It's going to be controlled so that you just perfuse an area when you need it. About 3/4 of the body's capillaries are actually shut down at any given time. So this is showing a comparison here. So you still always have blood going through this vessel here, but when you have the meta-arterial and these valves open and relaxed, you can see perfusion is happening. when the precapillary sphincters are contracted, it isn't there isn't blood in the area, it's just not going to perfuse to the tissues. So as you go through the different types of veins, the structural changes are not as distinct as they are in the arteries. They do have relatively thin walls in relation to the total diameter. They do have the same same 3 layers though. So the tunica interna is thinner than it is in arteries. The tunica media is thinner and has less smooth muscle than it does in arteries. And the tunica externa is the thickest layer. So veins are not designed to withstand high pressure. So when there is pressure on them, it tends to damage them. The valves or folds on the tunica interna, they form cusps. They're important for venous return because venous return isn't a continual stream flow. There are surges in it because of muscle contraction and things from the heart. So this will prevent backflow in between those surges. So there's 3 categories of veins. The venules are the very small veins that are going to collect the blood from the capillaries. They'll move to medium-sized veins. They're going to have a thin tunica media and a few smooth muscles. The tunica externa is going to have your longitudinal bundles of elastic fibers. And then it will move on to the large veins that have all 3 tunica layers. They have the thicker tunica externa and thinner tunica media. So remember, those valves in there are made up of tunica intima. So what will happen is you have muscle compression that will help move blood towards the heart in the peripheral system. So here is a relaxed muscle here with the valve closed. Here, when the muscle contracts, it's going to squeeze the vessel, pushing the blood through. It opens the valves temporarily, and then when it relaxes, it will have the valves close again so the blood doesn't flow back down. This is showing the cusps of the valves inside of a vein. So where does all of this blood go? The largest portion of the blood is at rest in the systemic veins and venules. So this is 64%. About a third of the venous blood is in large venous networks of the liver, bone marrow, and the skin. So if venoconstriction is going to reduce the volume of blood in the reservoirs, this is going to allow greater blood volumes to go to other places where it's needed. When we look at the circulatory routes, the simplest and most common route for blood to go is from the heart to the arteries, to the arterioles, to the capillaries, to the venules, to the veins, and they only pass through one network of capillaries from the time it leaves the heart till it returns. So the blood will go to a location, it's going to drop off and do its exchange at that location, and then it will return to the heart. So that's the most common one. That way everybody gets their own fresh blood supply. Occasionally we will use portal systems where it will go through 2 consecutive capillary networks. So it's going to stop at one place and have some exchange, and then it will go to another place and have some exchange. This is specifically because we want to take materials or substances from the first location and deliver them to the second location. Most of the time, you don't want the location getting secondhand blood. But here, it's important that it gets the stuff from the second stop because you're delivering components from that first location. So the portal system is how we're going to do that. And we have these between the hypothalamus and anterior pituitary. We have them in the kidneys. And then we have between the intestines and the liver. So this is a visual diagram that lets you see some of the comparison of these different types of systems. So here down at the bottom, You can see where there is some anastomoses in here, where you can have the two vessels will merge. Up here, you've got a portal system where it's going to make one stop and then a second stop. So when you have an arteriovenous shunt, the artery flow is going to go directly into the vein and bypass the capillaries. So this is an example here of a shunt where it's bypassing. So we can have venous anastomosis here, which is the most common. One vein is going to empty directly into another. And then arterial astomoses, where two arteries are going to merge to provide collateral alternative blood flow routes. So the coronary circulation is a really common place to have this because it's so essential that we maintain blood flow to the heart. You'll also have it around the joints. So there's some different ways that we have capillary exchange occur. This is the movement between substances in the blood and the interstitial fluid. So one is diffusion. So you can have things go through the membrane. So this would be lipid-soluble substances that are able to do this. That are able to just go through the membrane. We can have transcytosis as well as bulk flow. So here you can see movement through intracellular clefts, water-soluble substances. Then you can have movement through fenestrations, where you've got bigger holes, and then transport via vesicles. So it's going to have to go into a vesicle. The vesicle will then cross and lead it over to the other side. So diffusion is the most important method. Things will move down their concentration gradient. So they're going to move from a more concentrated area to a less concentrated area. Oxygen and nutrients from the blood will do this into the interstitial fluid and body cells. Carbon dioxide and waste will move from the body cells to the interstitial fluid and then back into the bloodstream. So they can cross the capillary wall through those intercellular clefts, fenestrations, or the endothelial cells. Most of your plasma proteins are not going to cross except in the sinusoids. And then in the sinusoids, you can have both the proteins and even some of the blood cells leave. So this is important. This is what helps give the blood-brain barrier. You've got tight junctions because we really want to limit what goes from the bloodstream into the brain. So with transcytosis, you've got a small quantity of material. Here you've got substances in the blood's plasma that can be enclosed within those pinocytotic vesicles. They enter into the endothelial cells, and then they're going to leave by exocytosis on the other side. This is used for things that are large lipid insoluble molecules that can't cross the capillary walls any other way. They're too big. So with bulk flow, it's a passive process where you're going to have large numbers of ions, molecules, or particles that need to move together, and they're going to move in the same direction. So it's based on a pressure gradient. But diffusion is more important for the solute exchange. Bulk flow is more important for regulating the volumes of fluid in interstitial fluid. So filtration, this is going to be how you move things from the capillaries to the interstitial fluid. Reabsorption is going to go from the interstitial fluid back into the capillaries. So capillary filtration is going to happen at the arterial end. Capillary reabsorption happens at the venous end. Glomeruli, they're going to be devoted to the filtration. Alveolar capillary, they're devoted to absorption. So things that can influence this would be activity or trauma. So in the next video, we will start to talk about pressures.